Well, hello everyone. As David said, I'm Ryan Bernhardt, Chief Commercial Officer, and what a pleasure it is to be back together again in person after 2 long years. I want to thank SLAS for continuing to put this on, and really excited to see all of the innovation out on the show floor and hear some great talks about what's been happening across the life science community. Today it's my honor to introduce Biosero and talk a little bit more about who we are and what we do. Simply put, Biosero is a automation company that develops automation solutions to enable scientific discoveries. And today, the theme of the presentation is really about orchestrating your discoveries and going beyond the integrated workstation. In the life science industry, we're all trying to resolve the same set of problems. How do we move faster with less people and at a lower cost? The problem is today that discovering drugs, cures, therapies, and diagnostics takes longer and costs more than it should, and manual processes create bottlenecks, inefficiencies, and variabilities. Many companies have terminologies and initiatives for this. They call it next generation research or next generation development or next generation diagnostics or industry 4.0. But all of these initiatives are really trying to resolve the same set of problems. How do we get innovation to the market quicker? Some of the historical causes are really largely around human-dependent variability. Anytime humans are asked to do a task, there's differences in technique, there's differences in the way we approach it, there's timing variabilities as it relates to how long an experiment takes, when did a cell culture get fed, when did it get split. These variabilities are largely around the human-dependent processes. With this, there's also only so much that we're able to just physically capture in regards to the data. When we're doing something on the bench, we're primarily focused in the sample, the experimental result, and the actual process that took place. But there's so much valuable data that is oftentimes left behind. There's also a lot of disparate data being generated in the laboratory today. We have isolated instrumentation, one instrument not talking to another. And overall, I would say the laboratory is one of the few places where we have limited laboratory analytics. And in most other areas, we know how much an experiment costs, we know who's working on the project, when it will be done. But in the laboratory, overall, there's really limited visibility or transparency as to what is actually happening. And this creates a very much a siloed environment. So one researcher may not even know what the researcher at the bench or the fume hood next to them is working on. You have a lab next to another laboratory that doesn't even know they're using the same type of instrumentation. And so you have these siloed environments, and overall there's a real challenge in lab connectivity, and ultimately this leads to inefficient lab productivity. And it's not like lab automation hasn't been tried in the laboratory before. It largely has, but I would say up to date, it's been rather mediocre in the success. There's been some real success stories, but there's also been many failures along the way, or laboratories that aren't getting the productivity out of their automation investment that they would expected. I've heard researchers say that the automated instrumentation in their laboratory is complex, it's intimidating to use, it's expensive, it's time-consuming to evolve it into something more than what it was brought in for. I've heard it be referred to as unreliable. And I've actually heard researchers say, that investment in automation was a waste of money. So Biosero had come at this challenge from a different angle. Biosero started with a different approach to the integrated workstation. Biosero, rather than coming at the solution from a hardware-centric point of view, as many other companies have done, where they started with a liquid handler and they had a software developed for that, and then along the way they said, wouldn't it be nice to add a washer and a reader and an incubator where we can do automated ELISAs? Biosero stepped back and said, let's come at this from what's best for the process. We need a process-centric solution where we'll use the best instrumentation or the instrumentation that a scientist needs for their process and allow to evolve over time. And so Biosero's flagship offering was Green Button Go Scheduler, which is a automated scheduling solution for integrated workstations. As you can see in the screen here, this is our layout tab. It allows scientists to build a digital twin of the workstation that's in their laboratory. Once they've created the digital twin, they can then move over to the process tab, and it's been designed as a drag and drop, low code, no code programming environment where basically if a scientist can talk through their process, it's simple enough to drag those corresponding steps to create that process in an automated format. Once the process has been built, the scientist can then queue that process up on a schedule, or they can run it in real time. But they can say, I'd like to run this experiment at 8 a.m. on Sunday morning, schedule it, and then launch it. Once the experiment is launched, you then have access to real-time monitoring that allow you to see everything that's happening on the workstation. As you can see here, the devices are lighting up in green. That means they're actually being used. Gray would be they're sitting idle. Red would indicate an error on an instrument. And yellow would mean that a scientist came over, locked the device so they could use it as a standalone instrument. On the right-hand side, you have a Gantt chart of the process and every sample plate that's running through that process and where it is. And then through this tab, we provide a set of analytics that allow you to visualize other things like utilization, error rates, the time a sample spent on each instrument to provide you a little more transparency as to what's happening on your integrated platform. As part of the Green Button Go Scheduler evolution, one of the things that we've done is our Green Button Go Scheduler is built on what we call a plug-in framework. Essentially what this means is we can continue to add new capabilities and through what we call extensions. Essentially, extensions are, they extend the core functionality of Green Button Go Scheduler for application-specific processes or customer needs. Here's a look at 5 of our common scheduler extensions. One of those is called Batch Planner, which handles the kinetic assays. So if you need high kinetic imaging, kinetic reading, our Batch Planner extension and it seamlessly enables that. Another one is called Cell Maintenance Planner. This automates routine processes that are commonly used in cell culturing applications where you may need to do a series of cell feedings over each day, and then you need to do a viability, a confluency measurement, and then spawn into a splitting application, and then go back to feeding until ultimately you do an endpoint analysis. This would take thousands of lines of code to program, but with this extension, it's done in a few clicks in a graphical universal interface to enable that and calendar up that over a 24-day period, 30-day period, 90-day period on these longer automated workflows. Our multipath scheduler extension is an extension that allows for the simultaneous control of multiple robotic arms or transportation mechanisms. It allows these arms to be used simultaneously, but also in coordination with one another to really increase the productivity and efficiency of your workstation. Our assay scheduler is a unique offering where it's actually the opposite of a high throughput screening environment. It's built to enable high mix, low throughput applications where when you invest in an automated workstation within your lab, you may have 10 scientists who want to all be able to utilize that system, and they need to be able to utilize that oftentimes in parallel with one another. And so Assay Scheduler determines optimal times to run your processes in coordination with other processes running simultaneously. And then our 21 CFR 11 Manager is for those laboratories that need that extra traceability and user access control in more stringent environments. In addition to our flagship offering of Green Button Go Scheduler, Biosero has developed a world-class integrations team where we really bring complete visions to life from beginning to end. And so a scientist may have a need to be able to leverage a automated high-content imager, but do it in a more walk-away fashion. And so we develop automated reader feeder type of workstations, but we also have a team that goes well beyond that to the BSL2 enclosed integrated workstations that have multiple robotic arms, 50 plus instruments connected together. We also do integrations on instrumentation that were really never intended to be fully automated, such as the integration of the LCs in this workstation. Today we have a driver library of over 400 unique devices. We have a dedicated driver development team that is adding to this on a weekly basis as new technology and new instrument arrives. But over time, there's really grown a need to go beyond the integrated workstation. And this is what we refer to the lab automation continuum. The reality is that although we started here and we believe we offer a superior solution to integrated processes, the industry has moved to needing a solution that goes beyond that. And when you think of going beyond the integrated workstation, you may think of the lights out, fully automated biofactory that's shown here on the right. But actually going beyond the integrated workstation is more than that. It includes being able to automate semi-automated or standalone instrumentation and being able to digitalize manual tasks that are happening in the lab as well. At the foundation of this continuum is data capture integration, meaning we want to be able to capture data in a manual process that has to happen at the bench or in the tissue culture hood in the same manner that we would capture that data at the integrated workstation, or better yet, in the fully automated lights-out biofactory. And so what does this look like in the lab? Well, for example, in this workflow, we have some sort of compounds needing to be picked. These are dry compounds. They need to be picked out of an automated store or a manual store. They're then taken over to a balance or a weigh station at the bench where a certain amount of neat compound is weighed out into another set of tubes. That tube is captured with a certain weight. Then that dry compound is taken over where it's solubilized with a certain solvent of interest at a desired concentration, and then that compound may be walked over to a workstation where we have the ability to create assay-ready plates, and then that assay-ready plate is taken to TCAN Fluent where it also meets up with a cell culture plate that has been grown and conditioned, and now the assay is performed at the liquid handler, where then finally that assay plate is taken over to a high content imager or a multimode reader or a flow cytometer or an LCMS. And so that may be an example of a workstation, but although this is the primary path, what's not being shown here is all of the touch points between humans in the lab that need to prepare reagents, buffer solutions. They need to get those in proper formats to then load on the workstation. You also have all of the consumables that need to meet up at each one of these steps. You have the parameters that need to be put in to the various steps within the workflow, and then you have all of the conditions that are happening in these labs or multiple labs in terms of timing with someone at lunch, and so this didn't happen for 30 minutes longer than it should have. What are the environmental conditions? All of that is happening. Wouldn't it be nice to have some way to manage this or control this, or better yet, we need some level of lab orchestration. And so what is lab orchestration? Lab orchestration can be defined as managing workflows by harmonizing processes, people, data, business rules, and materials to achieve desired results. You see in this diagram here, we have all of this coming in where it's being harmonized, and then out of it comes results. And those results may be knowledge. Those results may be that next blockbuster molecule. It may be a better process, a more efficient or cost-effective way of reducing the time of that process or reducing the amount of material used. It could be a new diagnostic that offers a life-saving test. The reality is that the results coming out of here, though, should be captured as knowledge, which can go back into the process to optimize that for the next iteration in the cycle. And so as part of this Biosero has launched our newest product offering called Green Button Go Orchestrator. And Green Button Go Orchestrator is all about being able to orchestrate not only the samples and the experimental process and experimental result, but Green Button Go Orchestrator orchestrates the flow of physical material throughout every step of the way. Green Button Go Orchestrator is an end-to-end laboratory workflow management solution. We're also orchestrating the flow, the digital flow of data. So in this example, you have data coming from a LIMS or an ELN that is corresponding to the samples that are running, but there's also experimental parameters that will be passed in this case, to this workstation. You also have all of the information about the consumables that are needed. So what type of labware, reagents, building blocks, tip types are being used. All of that's captured and then corresponds the physical action of getting that labware to the workstation, where then you have the execution of the process at the workstation level. And as part of that execution, there's a ton of valuable data being generated in real time that we're able to capture. So we can capture information about the seal type that's being used on the sealer, the lot of the building blocks that are used, the number of times that a cell line has been passaged prior to being added into that assay plate. We can capture information about the temperature and the humidity in the laboratory. We know information about how long did that assay plate sit out of the incubator before the read happened. And we're able to capture and contextualize all of that data in coordination with the control of the process, where we're able to harmonize that into a single location. Now, this can happen for each step in the process, and although it's showing 3 automated workstations, you could substitute any one of these workstations for a scientist at a bench, a standalone liquid handler, or a standalone LCMS that could be tied into this orchestrator ecosystem. And the idea is that we can provide all of that data back and make that available to integration with LIMS, ELN, inventory management system, asset management system. Maybe you have an AI machine learning algorithm or a retro-experimental analysis tool that you want to be able to use, because all this, although we're showing this as a linear process, we all know that the scientific process is anything but linear. It's an iterative cycle where we learn and we look to improve based upon the results of what we've done previously. And so Green Button Go Orchestrator really began out of the work that was done with the Lilly Life Sciences Studio. This orchestrator was designed to oversee the integration of 22 autonomous workstations that are connected by over 200 meters of MagnaMotion track. Orchestrator was built to control the flow of the physical materials that were on the being used on the system, but also the bidirectional flow of data that was coming and going to the system. The other aspect of this is that there's data being generated in real time that is pertinent to the next step in the process. And so Orchestrator was built to be able to capture that data and make dynamic decisions about what to do next in the process. And although this concept originated here, Orchestrator has been commercialized and has greatly evolved beyond this to a productized solution. You can think of Green Button Go Orchestrator, it's a software suite. So you can think of it as kind of like a Microsoft 365, and the Orchestrator software suite is comprised of 8 applications, and these 8 applications are used in the planning, scheduling, execution, and optimization of these end-to-end laboratory workflows. These are the 8 apps, and I'll be going into a little more detail on these, but we have an app called Data Services, Insights Dashboard. Green Button Go Scheduler is actually one of the apps within the Orchestrator software suite. Workflow Designer, Workflow Conductor, Transportation Manager, Lab Experience, and our IoT Instrument Agent. Now, just to give you a view of the architecture of Orchestrator, I want to start with this diagram, and I'll bounce back to this diagram throughout the remainder of the conversation. At the center of the Orchestrator architecture is data services. We refer to data services as the heart of Orchestrator. Essentially, it is data warehouse that is connecting all of the other Orchestrator applications together. Data services also captures other resources within the laboratory and handles bidirectional flow of data to your internal or external software tools, such as your LIMS or your inventory management system or your ELN. But data services is really that hub that keeps everything connected within the lab. And this is the role of data services is really all around the contextualization of data. So in this example, what you see is data being generated, and in this case, it's 24.65 degrees Celsius. In and of itself, that captured data really it doesn't mean much. But as we begin to apply context to that piece of data, all of a sudden it becomes information, it becomes more relevant. So when I say that 24.65 degrees Celsius is a temperature coming from this incubator with this serial number at this date and time, now all of a sudden I can apply some level of knowledge to that and say, well, for that incubator, that temperature's out of range. And once you have that type of knowledge, you can then apply action to it. So in this event, I'd say this temperature is out of range for that incubator. We need to call a service technician to have them come in and service this. The value and the comprehension of data as it's contextualized increases drastically, and that is the role of data services as that centralized data warehouse that we want to be able to capture and contextualize all of that valuable data into that single location and make it available to the entirety of the Orchestrator ecosystem. Now, this is another representation of data services. Data services is kind of being shown here in the middle, but the reality is that we're capturing data from everywhere and making it available to go anywhere. In this case, these are some common sources of where we're streaming data. We refer to every action that happens as an event, and that event is streamed. So we're using the same technology that's used in social media, whereas if I like a post on LinkedIn, it sends a notification to my whole community that I've liked a certain post. That action of a like is an event, and that event is then captured and streamed. That is the same technology that we are using in the Orchestrator ecosystem to say everything that happens at a workstation or at an instrument is an event, and that event is automatically streamed into data services. Once it's captured in data services, it's stored, and now that data, that contextualized data, is made available for numerous modes of presentation. So this could be a monitoring dashboard, this could be mobile notifications, this could be business intelligence, or maybe you just want to use it for some forensics analysis. The reality is we don't know the questions that you're going to have, but we want to make sure we have the relevant data in place to answer those questions that may come up tomorrow, next year, or even 5 years down the road. Data services is then at what we call the heart, and the role of data services is to connect these other components within the laboratory. So in this example, I now want to just briefly switch to what's being shown here is we have two instances of our Green Button Go scheduler. These instances of Green Button Go Scheduler are on two separate workstations. So Green Button Go Scheduler, as we've already talked about, is the application that allows the scheduling and execution of processes at the integrated workstation level. Data Services is what is able to bring those two separate, disparate workstations together so that they're working in unison with one another. Data Services is handling that digital capture. But what about going beyond the integrated workstation? What happens when you have that standalone liquid handler, the standalone LCMS, or the NMR that's in another laboratory? How do I connect that? And that's handled through what we call our IoT instrument agent. So our IoT instrument agent is what allows us to operate and track processes and data for standalone equipment. In this case, you're looking at what you saw there was the uptime of that instrument, and here you're seeing the instrument availability for when that instrument can be used. We can capture and monitor utilization of each instrument within the lab so that you know how much it's being used. Should I invest in another one? We can also tie in other sensors and IoT devices where we can stream the humidity in the lab, the temperature in the lab, the luminescence in the lab. But in this case, not only can we digitally connect these instruments, but we also now create the potential to physically connect standalone instruments within your lab so that when a scientist interacts with that instrumentation, it knows all of the information about that sample plate that it's just scanned in. In addition to integrated workstations and instruments, how do we capture information or a process that had to happen at the bench or in the fume hood or in the tissue culture hood that's having to happen with a scientist or a technician? This is where we are able to use our lab experience application. The lab experience application essentially digitalizes SOPs such that it is enabling the capture of data that's happening manually. This is a bidirectional flow, so you can actually have these SOPs that are guiding the scientist as to what to do next. We've integrated with tablets and smartphones, audible technologies such as Amazon Alexa and Siri, but we've also integrated with technologies like the HoloLens, where you can visually capture and visually see what's happening in addition to audible recognition. These are some of the screens that have been created using Lab Experience. Lab Experience has its own workflow builder, which is a drag and drop, and you have the ability to create your own user interfaces. You can use it to digitize routine maintenance protocols, to instruct someone in the lab as to how to perform those. You can digitize inventory levels and then set alerts based upon when the inventory of a certain number of plates becomes too low. But we have what we call the Lab Experience Builder, and it gives you a graphical interface to actually build out your protocols, and then you have complete control over the user interface that's being generated. The whole idea is that we want to create a frictionless environment between SOPs and scientists and enable a bidirectional flow of data that instructing the scientist what to do next while capturing the data as it is performed. This reduces the variability that comes from person to person, technique to technique. They're being instructed what to do. But we also wanted a customization of this because we recognize that the experience in your lab may be different than the experience in my lab, and you need to build this out so that it's your lab experience. So now we've captured the integrated workstation, we've captured the standalone instruments, we've captured the manual tasks with lab experience, but what if we want the ability to automate the connection of these things? So we want to have materials moved automatically or simultaneously between these. This is the purpose of the transportation manager application. So a laboratory can start with digitally connected systems, and over time they have the ability to add an automatic mechanism. And I should highlight, the reason this is shown in this architecture is Green Button Go is modular. It's what we call a Lego block-like architecture. So you can mix and match these different applications for your project or your process, and then over time you can add different applications to that. You could scale it. It's not a one-size-fits-all, and so this gives a lot of additional flexibility for this product to grow as your portfolio changes or as your needs grow. And so this is our transportation manager application. In the L2S2 video, it's what is being used to control the MagnaMotion track to connect the disparate or autonomous workstations. We've also enabled the use of mobile robots as part of the automated transportation manager. We have other conveyor belt systems and other independent cart technology track systems that we have implemented. And more recently, we've had a few customers ask us about the ability to use drones to move materials from one workstation to another. And although we haven't done that to date, it's totally possible within the architecture of our Transportation Manager app. And so in this example, you're seeing the ability to control MagnaMotion track. We can control various robotic arms, mobile robots, and other conveyor systems. And the idea is that the vehicles that we're using to move materials within the lab are going to greatly change and evolve over time. There will be a new technology that comes out and that's better suited, and we'll be able to enable that. The other thing I want to mention is with this transportation manager, it no longer means that you have to have workstations or workflows that stay within a single laboratory. With the use of mobile robots, we can now connect workstations across multiple laboratories. We can control elevator systems, automatic doors, and so it begins to expand the capabilities of these automated connected systems. So now once we have the ability to control processes at the integrated workstation, we have the ability to control processes at the instrument level, processes at the manual level, and we have ability to connect those components together with Transportation Manager. How do I go about building my end-to-end workflow for my laboratory? And this is the role of the workflow designer. So workflow designer, much like Green Button Go Scheduler, offers a drag and drop, low-code, no-code environment that allows you to easily build these end-to-end workflows. But now instead of building processes at the workstation level where you're connecting instruments, now you're building end-to-end workflows that are connecting processes at various workstations and instruments together. What this means is you have the ability to connect multiple automated workstations together, or standalone instruments, or any combination thereof. In this case, you would register your process at the workstation level or the instrument level. Once that process is registered, it now becomes available within Workflow Designer, where then you can simply begin to build these end-to-end workflows. What you're seeing here is using a sample aliquoting process at a workstation. We've then incorporated the transportation step, which in this case may be a MagnaMotion system. We then have the ability to add in the extraction process at the extraction workstation. As you notice, you can still use looping functions and all your normal if-else programmatical languages to create deviations in your workflow. But you have the ability to then, in this case, we're adding an amplification process at another workstation, and then finally transporting that to the endpoint analysis. Once you have designed your workflow, now how do I actually launch it? And that is the role of Workflow Conductor. So Workflow Conductor gives you the ability to launch workflows from anywhere in the world. You can launch it from the laboratory you're in. You can launch it from a meeting room. You can launch it from a soccer game, or you could actually launch it from somewhere else in the world and have that workflow started. In this case, you're able to define the variables or the inputs that you need as part of that end-to-end workflow process. You have the ability to calendar that up and say, I want to start it at this date and time. And then you also, once the order has been launched, you have the ability to see every order that's running in your process. When you click on that, you have a full field of all the details about that workflow. Who was the submitter? When was it submitted? What state is it at? But you also have the ability to see everything historically that is run and whether or not that's successfully completed, and numerous details about that. And so this is a look at once you've designed your workflow, now they're available to call up from anywhere in the world. You can then launch those workflows. You can enter your parameters that you specify for those workflows. And then once that order is created, you have the status window that allows you to see active orders. It then calendars that on the calendar so you can see when it's running in your laboratory. And then once it's officially launched, you'll be able to see that actually on a workstation running and where it's at in the process. And so that really brings me to the final application within the Orchestrator software suite, known as our Insights Dashboard. The Insights Dashboard is what we refer to as the air traffic control tower of Orchestrator. This is what allows you to see everything that is happening across your Orchestrator ecosystem. So you have a real-time monitoring view of everything that's happening. and the status of your workstations, but you also then have historical analytics that you can use to improve the process of your laboratory or of your workflow. And so in this case, this is showing a real-time status view where you see the workstations that are running, you see the progress of the particular process that's on that workstation. This real-time view also shows you whether or not a workstation or an instrument is in error. It gives you the ability to remote into that to address it. It shows workstation utilization, instrument utilization, heat maps. You also have all of your sensor data built in here, and you can also tie in to the Insights dashboard all of your internal or external software resources. So if you're tied into a LIMS system, you can actually have a button or a portal that the Insights dashboard becomes a portal to your LIMS system or to your asset management system or your inventory management system, where it creates kind of a one-stop shop for being able to oversee the operations of your laboratory. Now, Green Button Go Orchestrator, what is the value proposition of this? Well, what it's allowing for scalable, automated, integrated solutions. In our industry, the paradigm is changing. Scientists are wanting more automation, more connectivity, but also more flexibility. We need to be able to take a platform and string together multiple modules or workstations that can operate and impact the portfolio independently, but also in coordination with the other tasks that are happening. And so Biosero is enabling these larger integrated workstations to be built where they're all overseen by Green Button Go Orchestrator, but each one of the workstations is running autonomously or independently with the use of Green Button Go Scheduler. We can also tie in instrumentation that may never be physically connected. And in addition to making these platforms these large connected systems, we can also enable large platforms that are not physically connected through the use of mobile robots, where we can now tie in a compound management center that may be generating assay-ready plates on the first floor, driving that up to an elevator to deliver at a high-throughput screening facility that may be on floor 4 of a research building. The other thing that the other aspect that Green Button Go Orchestrator is enabling is this idea of the globalization of lab automation solutions. Our world is more connected than ever before. We have colleagues that are in all parts or different parts of the world, but we may all be needing to work on the same projects and the same automated solutions. So this orchestrator framework allows us to have global connectivity and standardization of automated scientific solutions where colleagues can do something in one part of the world and seamlessly roll that out to colleagues in another place around the world. In this example, we're actually doing this currently with our Biosero's Acceleration Lab. So in this case, it's a global connectivity showcase of the world's most advanced laboratory orchestration concepts. We started with an acceleration lab in San Diego, which really was a place where you could visualize our software solutions in actions. It's one thing to talk about software, it's another thing to see it in action, and that's exactly what we built this for, was the ability to demonstrate this. But we also built our acceleration lab to be a innovation or testing grounds for new technologies. And this has really aided in the evolution of our product offerings and the innovation that scientists are getting to leverage within their laboratories. Today, we've expanded it to an acceleration lab in Japan. We're currently building one in China, and we have acceleration labs underway in Boston as well as in Freiburg, Germany, where we can now stay connected to these different laboratories around the world and pass methods and workflows across to these laboratories. We also have the ability to monitor all of these different sites from the Insights dashboard. So the markets that Biosero is primarily a part of today is the pharma, biotech, clinical diagnostics, agriscience, academia, and government. We're implementing automation for applications ranging from synthetic biology to synthetic chemistry, to QA, QC manufacturing environments. And one of the unique things about Biosero's software offerings is that these tools are being used in discovery research labs where you need a great deal of flexibility and agility, to development labs, to CLIA-certified clinical diagnostic labs, and even GMP, QA, QC manufacturing labs where you have the most stringent conditions. And a big part of Biosero's focus is on customer success. We have a motto that your success is our success, and that's been a key to every customer that we work with is considered a partner, and that partnership is really what has enabled the evolution of our product offerings and that focus on innovation, because it's what's best for our customers. And so with that, we have designed automation solutions specifically for scientists, not just engineers. And with this, we've created solutions that are process-centric, that are seen as adaptable, flexible, that are easy to use by scientists within the lab. But these systems are also powerful and they're supportable. And the goal of this has been to provide a level of transparency through the capture and access to data that has really not existed to date. And the idea is that we do this through a harmonized solution where we're able to design solutions for scientists to enable the orchestration of your discoveries. And so with that, I want to thank you for your time. I want to also give a short plug for Biosero's Acceleration Lab and invite you to please come visit our Acceleration Lab tour, whether that's in person in San Diego or coming soon. There'll be one here in Boston. But you can also join virtually for a live virtual tour of our Acceleration Lab, where you'll see all of Green Button Go Orchestrator in action. And so with that, thank you very much, and looking forward to hearing from Jesse and Andy.
Hi everyone, I hope you can bear with me. I have two people at home that are immune compromised, so I'm going to be keeping my mask on. My name is Jesse Mulcahy. I work at EMD Serono in Billerica, Massachusetts, so we're local. I've been working with Ryan for, let's say, for over 10 years across probably 6 different companies between us and things like that. It's been a great journey. So EMD Serono's U.S. research and development hubs in Billerica, Massachusetts. We support our oncology, immunology pipeline. Last year, we completed the construction of our new state-of-the-art R&D facility. The building has 145,000 square feet of mixed laboratory and collaborative working space that can accommodate up to 400 employees. And here's actually a picture of one of the systems that we recently built and things like that. It's an absolutely beautiful campus. If you'd love to come see it, just drop us a note. The bioassay automation and compound logistics department provides automation and screening technologies to support the advancement of all NCE and NBE discovery projects. Leveraging BAA's expertise in robotics, we are working across discovery technology to modernize our practices and adopt automation. Our robotic liquid handling instrumentation allows scientists to save time during assay development, train new personnel faster, free up resources for data analysis, and streamline assay transfer initiatives. In a discovery setting, flexible robotic solutions are also important to ensure that automation meets the business's current and future needs. We are implementing generic solutions that combine automated liquid handling, robotics with integrated instrumentation to achieve walk-away assay support. Over the last few years, the site has made significant investment in modernizing our processes. We have spent over $4 million to bring in standalone liquid handlers, modular systems, and fully walk-away work cells. With the support of our site leadership, our responsibilities have expanded from supporting single department automation needs to the entire site. To ensure easy adoption, user-friendly graphical user interface interfaces enable the systems to process many different assay-specific parameters. Operator intervention is also reduced by utilizing scheduling functionality available within the automation software to control integrated peripheral devices. To accommodate different study designs, work lists specify custom dilution volumes for controls and samples. The most overlooked benefit to these systems is they are built in a generic modular manner so that code and best practices can be shared across all systems. For example, our Hamilton training, when we implement a new system, we have a 2-hour virtual classroom training that we offer all of the operators, users, and things like that. We follow that with a 2-hour small group hands-on training that's about half of it is generic to running a liquid handler, then the other half is dedicated to the specifics of their unique system. And then we also follow that with a 1-hour hands-on training one-on-one. And what we've seen is Hamilton's actually, vendors are really happy because we're not breaking things, strangely enough. But that comes from the competency of our users and things like that. And because some of our systems don't have redundancy, putting in the work to make these systems, as user-friendly as humanly possible really saves us a lot of headaches and things like that. We have, even though we're not regulated, we have SOP quality job aids that are, again, written generic, so we can share them across all of our systems to train everybody, to get everybody 80% trained, so then that last 20% we can customize to their unique solution. The other thing that's nice is our user interfaces are also harmonized across our systems. So if we have one person that's trained in on using a liquid handler for cell-based assays, getting them up and running on another system for instance, protein purification, or you have an LAL assay automated, it's really quick and easy, and things like that. Our original work cell was designed for acoustic dose response curve generation. As you can see, the system that was delivered at the end of 2018 was a very robust work cell utilizing MOXA serial to Ethernet device servers and advanced tech Atom input-output modules for controlling drawer and table sensors. Unfortunately, after a global alignment initiative, our strategy shifted towards tip-based DRC generation, and the need for acoustically generated serial dilutions diminished. After reevaluating our needs, we determined that the system would be better suited to assist with our phenotypic screening assays and more complex routine work. This meant that our system needed to undergo significant modifications to support these new requirements. We approached multiple integration vendors to discuss the upgrades that our system needed. Because our system would be constantly evolving, we needed an integrator that would maintain the existing hardware communication infrastructure and also allow us to perform our own integrations. Biosero seemed like the best fit based on these requirements. Because of the freedom we had to perform our own integrations, we were able to add a modular cart location that utilized docking brackets designed by our HTS systems in Germany, right here. I think it's like $200, $400 worth of hardware. It's based on a door latch that you see typically on a screen door. And it's really great because all of our carts can be on wheels. We lock the casters, and we get phenomenal precision and accuracy with robotic arms. Another complex aspect of this project was our desire to have a tight integration with the Hamilton Starlet's Venus liquid handling software. We wanted to use a scheduler to pass all the plate and pipetting parameters to a single Venus script, which would allow us to avoid using dozens of separate Venus scripts to achieve the same level of flexibility. This allowed the Hamilton Starlet to control the order in which specific plates are passed to the system by the scheduler. So GBG creates a work list at the beginning of a run, and that's sent to the Venus system. Venus consumes this work list and then creates arrays of all the information, so that as we're processing plates and things like that, you get a two-way communication between the Hamilton software Venus and GBG, where Venus is taking over and telling GBG what to do. So Venus is telling the GBG what plate barcode to pass and things like that. It works phenomenally well. And I'd actually like to thank Stephen Damon and Prabhakar Henry. They were just amazing on this functionality we implemented. We also wanted to use incoming and outgoing handoff nests to queue up plates as needed. This required using custom Hamilton sensors to monitor the handoff nets and avoid traffic jams. This decision tree right here, just these two decision trees showcase the complexity, how complex of a process we had to make to then actually make it very simple. And so the Venus is monitoring the handoff nests right here, so we don't have the eye swap on the Starlet giving a high five to the precise arm, which happened a couple of times until we modified the hardware and the timing and things like that. Another custom solution that we implemented is our Access Workstation. Because of past difficulties with alternate centrifuges, we wanted to use a Bionex High G3, making our Access Workstation a non-standard build for LabSite, now Beckman. Fortunately, LabSite was willing to allow us to integrate the High G3 on our own. We now have a very robust centrifugation option in our system and are standardized to the same centrifuge across all of our work cells. So the funny thing is we're working on our GBG system at the same time as the Access Workstation, and we ended up swapping the centrifuges from one to the other because we had I think there was a delay in procurement, or we had a hardware issue, I can't really remember. But it was nice that it's great that we're able to align our hardware across all of these systems because then we have redundancy and things like that. Another driver that we worked with Biosero to develop is the Echo Cherry Pick driver. We wanted to align our file handling across our Access Workstation that I previously showed and our GBG work cell. This allowed scientists easily to scale their assays from one system to another while also using the powerful capability of the native cherry pick application within the Echo software. As you can see, we went from needing the reformat and cherry pick app to solely using the cherry pick app with control and sample source plates functionality. The curly brackets you can see, well, it's hard to see in the work list, but the curly brackets are used to identify regions as identical well content, reducing the issue with well depletion. The colon parameter allows for regions to be copied from source to destination. This may not seem like a big deal, but we took a work list that was originally 595 lines and cut it down to 275 lines by using regions, and also we removed the DMSO edition from the Echo to then implementing a Combi NL dispenser in the process. Just overall, we were able to save 3 minutes, 3 minute savings per plate. This became critical because while we were automating this assay, we were using a dummy colorimetric read for our Envision. Didn't really think any big deal. We got everything automated. It was great. Then once we implemented the Nanobret protocol on the Envision, the read times were really extended, and that actually caused issue with the stability of our substrate. So we had to look to see what we could do to optimize the assay as much as humanly possible to fit in as many plates as we could within that substrate stability time. This is our GBG IQ work cell, so we're able to source all these components for the system on our own, and then work with a local vendor to assist with the design build. By aligning the hardware with our first work cell, we're able to implement a hybrid system that was aligned with our HES strategies in Germany. This system will be used for high throughput sample preparation for FACS, using the IQ screener to support the MAB discovery pipeline. Beyond FACS, the system will also support the functional assays during early MAB screening with our integrated vision reader and IQ screener as a readout. So this is a system, so we had a phenomenal opportunity with building a new building. There was a CapEx funds made available at the very last minute in 2020. And we were able to build the systems ourselves, working with another vendor that helped us to basically source the components that we identified. And I think we ended up building this for like half the cost of what integrators typically charge. And for us, it was a situation where in September, they asked us if we could spend a significant amount of CapEx. And this allowed us to get everything we need and then also have the flexibility for future work and things like that. So we're not locked in. And what was great is all the communication and so the communication, hardware, turntables, drawers, everything is exactly like our first system. So with, you know, that knowledge transfer and things like that, we're training people up on one system and they're going to be able to work and support the other. So for us, what we have coming up, you know, future, what's next? We're evaluating right now high throughput crude compound purification work cell to hopefully impact the design, make, test, analyze cycle. We're also looking at cloud-enabled incubator monitoring for monitoring the temperature, humidity, CO2, liquid nitrogen, DOR, water levels. We're working with a company called Larian right there. And also, we're looking at orchestration software. So we currently have work cells in, we have 5 work cells in Darmstadt, Germany that are used for, 3 are for HTS screening and 2 are used for compound distribution. And we're looking at using orchestration software to tie in all of these different work cells that we have around the world so that we can then monitor, you know, through a dashboard, monitor instrument usage, capacity, things like that. These are some of the people I'd like to thank. You know, from Biosero, I really want to make sure I thank Darren Arney, Prabhakar Henry, Stephen Damon, Dave Damon, Tom, Andrea, everybody. It's just been a great partnership for us. And so, yeah, thank you very much.
Good afternoon, everyone. So I'm Andy Smith, and I'm a senior automation engineer at AstraZeneca. I'm going to do my best to keep the time because I know that there's going to be questions, and I know there's a keynote at 1. So I'm here to talk about our robotics and automation development group, which is what I sit in, and how we're trying to build and design integrated both walk-up and lights-out solutions. So I'm actually going to jump in where Jesse left and say one of the most important things we recognise here is these things. It doesn't just take a village, it takes several villages. In this instance, we're going to talk a little bit about the AstraZeneca folk in this POC and a long list of Biosero, but obviously also a number of vendors. There's a much more extended list than this, so this is really just a focus on the slides today. So within the Robotics and Automation Development Group, or RAD team, what are we there to do? So what we're looking at is designing and integrating robotics and automation tools. And this is a theme that's been brought up already. These are tools for scientists. We are an incredibly lean team. We essentially support about 100 people per person. If we were to do this as a service, it simply wouldn't work. So we're multidisciplined. We come from a variety of bench backgrounds, but we would include things like the high throughput process development. Up there's the Tecan Evo running a RoboCon experiment, and that's something we're looking to publish this year, so watch out. There's going to be something interesting coming out there. My background's really in the high throughput process analytics, so we've got a stunner, traditional robotics, so Hamilton Star there, as well as something we're branching into now, so the more things like the storage and sampling and tracking. So we have the Verso Q20 that we've just brought in as well. I'm sure most people recognise this, but both the automation emphasis and interest has grown during COVID for obvious reasons. So another thing that we've folded into this is the remote support and monitoring aspect. So we have things like TeamViewer as well as cameras to monitor our tools. So, reiterate this one more time. It is critical that we view automation as a modular tool and that we're doing this for all scientists, so it has to be accessible. We need to deal with the aspects that Ryan mentioned about these things being intimidating and these conceptions about them being unreliable and issues around them. So I'm going to talk a little bit about our journey. As with all these things, there's lots of acronyms, and one of the things also critical is to think about the terminology. So when we say we're on this automation journey, we have things in flight from conceptual right through to where we think production. Predominantly right now, our production is our standalone liquid handlers, and we have some proof of concept and pilot things around the Biosero workflows. And what's important here is when we think about having users around 10, if this was a service, that would probably be the number of people supporting this as a service. So those tools can be a little bit more engineer focused because they don't need to be so accessible. So again, the way we're looking at this is when we think about walk-up, what we mean is that the slightly confusing 24-hour clock, but it's useful later on, is that if we imagine this idyllic world of working somewhere between 9 to 5, if that happens ever, but I don't think it does, we would be using these tools, we'd be walking up to them, using them as a standalone liquid handler, for example. They'd be operated within those business hours, and that would be the extent of that use. And essentially, people would come in and watch quite often more than we'd like. So it really is equipment that's utilised just during the personed hours, and clearly there is a big operational gap there, and we could do a lot more to fill that. So another way to look at this, and this is probably unfair because we're in winter in Boston, is think of it as an island of automation. So it's a liquid handler, it's fantastic, it does a lot of things to reduce our RSI, there's a lot of data available, but it is essentially an island. And this is where a lot of people in my team cut their teeth. A standalone liquid handler, fantastic. It does what it does, but it isn't going to get us where we need to be. I'm sure a lot of people in the room have thought about, well, we could add some data linkages. So we have, for example, a linkage with our labware LIM system, so we're able to pass our CSV files. Obviously, the Hamilton database in the background has all of the moves of the samples, so we're able to track and trace things going into our LIM system. You fold in a Cyanomics and some Hamilton decappers, and now you're starting to be able to think about a lab which can fill these tubes, label them, submit them to LIMS. This is great, but this is still what we would think of as Nike automation, as my boss coined the term. So you're having to move between these individual islands, which still is a massive benefit over anything manual, but it is not ideal. And this is really where we joined the Biosero conversation approximately 2 years ago now. And one of our first cart installations is the sample handling cart up there. So you can actually see co-located on the cart are the decappers, the Cyanomics tube labeler, and it's docked to the side of the Hamilton. And we have the high precision arm to move the samples around as well as a carousel there. So clearly this is an advantageous situation. You can move things between those systems, but we would still be really thinking about this currently as a walk-up solution, but it's kind of end-to-end, but it would still be during the worked hours. So the other terminology used in we're throwing around right now is the lights-out automation strategy. So this is really important that we are able to think about these processes as operating when there are no people present, so therefore they need to be robust. Obviously, we want to be able to monitor them and have various alerts, etc. And really, what we don't want to be doing is focusing this in a large work cell, a static system, and one that can only be accessible as a fully integrated solution. So where we are currently is in this kind of what we're calling the orchestrated lab up here. So again, we'd have our soon-to-be installed plate labeling solution, our sample cart docked to our Hamilton, thinking about the storage opportunities here, so bringing in the Verso Q20 so that we could aliquot these samples and store them. And this could all be scheduled and with the Biosero products. We'd also having Titian Mosaic into this mix so we can track and trace all our samples. And the thing that's going to make this really sing, obviously, is the Orchestrator suite of tools. And the thing that we're really, really excited about is the Omron mobile robot, because not all of these solutions are located in the same place. So the ability to schedule these tasks, have a transport manager, move these samples with the mobile robot between these work cells in different labs, hopefully in time in different floors, is a really exciting prospect. So just to focus on this one little part of the story, sample handling and the Omron, we've got our remote support monitoring camera mounted to our sample cart. So if I advance this, we can see this coming to the end of a process. We're going to pick our samples, we're going to put them on a scanner, and we're going to present them to a nest so they can be picked up. And just in the corner there, you can see Jarvis, our mobile robot in the corner. And as this moves, and we're going to move the mobile robot round to the handoff station, and what you can actually see happen is it goes from the dark lab, and as soon as that mobile robot moves, the lights come on. So we're able to demonstrate that this really is a valid way of thinking about using these work cells in a lights-out fashion. And this is probably the thing that gets the most attention, is the mobile robotics, the lasers, mobile arms, etc. And clearly we're all fond of this stuff. We're here at a conference, but really all we have to focus on is why it matters and what it's going to enable. And our aim is really to tackle some of the things that were discussed earlier, so removing manual tasks, low value added tasks. We have processes like our bioreactors and our workflows go on for 14 days continuously. There's weekend work, there's low value added tasks happening there, there's small sampling events, things are moved to various analytical pieces of equipment, to storage. So all of this would fit nicely in this idea of orchestrating all of these tasks. Again, we come back to training the scientists. We really want these to be scientist-friendly tools. It is not going to be on our team to solely run this. And one of the things that we're really proud of is we've published things like our high-throughput process developed scaled-down models, and we're working on some PAT papers as well. Again, these could be construed as islands, so we'd love to make this a continuous process and link those together. And we have this lofty ambition of a 7-year plan of lights-out collaboration and automation within the lab. And crucially, and it's good to see CFR compliance in there, is that we want this to go from beyond development and into the manufacturing space. So a quick snapshot of what our lab and the tools that we're talking about in terms of our high-throughput scaled-down models and our analytics would include Amber bioreactors, Agent LCs, Hamilton storage devices, both Hamilton and Tech and liquid handlers, our carts, and through to our storage. And again, as Ryan alluded to, these are not linear processes, these are cyclical, so there would be this idea of things moving backwards and forwards. It's also important to remember that we would still see these individual tools and workstations as walk-up, so during the worked hours, people can come and interact. So when we put those things together, we're able to tackle some of the concerns about robotics, really maximize our time, and have this kind of lights-out functionality, but still retain the walk-up and accessibility that we've developed in our scale-down models and some of our automated tools that we have available is not just standalone automation, but certainly things that are not necessarily designed to be automated. And we've got some really nice examples and hopefully some future publications and presentations to share. So I'll end on a much more comprehensive list of the folk at AstraZeneca and a quick shameless plug to say we are hiring automation engineers within our business, and I can see a colleague over there who also has a vacancy. So hopefully I've left enough time for questions and so we can get to the keynote.